My name is Jesus Villanueva Millan, and I am project scientist at MAST Program, and I am presenting a poster at DDW 2026, which is entitled Archaea are highly prevalent in the human small intestine, and the absolute abundance of Methanobrevibacter is associated with older age and greater microbial diversity. Archaea are part of the microbiome, but once they were underestimated. But now we know that they have beneficial and detrimental properties. For example, Methanobrevibacter smithii is associated with constipation and obesity, but also appears in more abundance in centenarians. Most of the studies are focusing stool, but we want to do this study to know what happens in the small intestine. And also we wanted to use absolute abundance to have a real idea of the potential biological impact of these microorganisms. To achieve this, we collected duodenal aspirates from human subjects that were in the reimagining study, and these subjects underwent EGD without colon prep. Once we got these duodenal aspirates, we extract the DNA and do shotgun sequencing, and then we calculated the absolute abundance of these microorganisms, the Archaea, in the human small intestine. 343 subjects were analyzed in this study, and one of the main results were that the most prevalent and abundant archaeal phyla were the phyla Methanobacteriota and Thermoproteota. And about the genera, what we saw is that the genus Allurubrum and Methanobrevibacter were the most prevalent and abundant. And when we wanted to see the abundance and prevalence of Methanobrevibacter smithii, that is one of the most known archaea, we saw that in the small intestine was the sixth one more prevalent and abundant, that the prevalence was 13%. Interestingly, we also saw that the genus Methanobrevibacter was higher in those subjects that were older, like from the 51 years old to 65, that's 66 to 84 in comparison with the youngest population. And when we compare this Methanobrevibacter, this genus, we saw that it correlates with the number of bacterial species. And the same happens with the species Methanobrevibacter smithii. So this means that there is more bacterial diversity in those patients with more of this archaea. So our conclusions are that the domain archaea, although low in total abundance compared to bacteria, is highly prevalent in the duodenum of these subjects. Methanobrevibacter genus is less commonly seen, but when present, we see its association with older age, and we also see an association with more bacterial species. That means that when you have more Methanobrevibacter, you have more bacterial species, so more diversity. Also, this absolute quantification provides a more rigorous evaluation of archaeal population and their significance biologically. Thank you so much for your interest in the poster, and also check out the other posters that we're presenting at DDW 2026.